Proteins are the bricks and peptides are the cement of life. Well, welcome to AHS Guide, your trusted companion for mastering the 5 core subjects of first year Allied Health Sciences as per Rajiv Gandhi University. Here we make complex concepts simple, clear, and exam ready, helping you learn smarter, remember longer, and build a strong medical foundation. In today's lecture, we'll focus on the biomedical importance of the peptides and the short chains of amino acids that perform a powerful function in the body. Our discussion will include glutathione, insulin and glucagon, bradykinin and angiotensin, oxytocin and vasopressin if possible, if there is. Okay, by end of the session, you'll understand how these small molecules, though the simple compared to the proteins, are vital messengers and regulators in maintaining the health and the homeostasis. So the biological importance of peptides is essential to learn because it is the building block of a protein. So when there is a building block of a protein, the body requires certain proteins to synthesize certain enzymes, hormones, or steroids. So proteins are the peptides are the basic foundation of building all these biologically important proteins. Now, peptides are the compounds which contain less than 10 amino acids and they perform a diverse biological functions in living organisms. So it just doesn't perform just one function, but it has various different functions which are essential for a normal human life. So the first peptide that we are going to study is glutathione. The type of amino acid present here is tripeptide with alpha glutamyl, cysteine and glycine as the amino acid groups present in the glutathione. How is it formed? By reducing and oxidizing. So the cell ratio here is approximately 100:1 for GSH:GSSG. And the function of the glutathione is it acts as a coenzyme for PGE2 synthesis and glyoxylase, and it prevents oxidation of sulfate groups, and maintains the protein and enzyme activity. So it is going to prevent the oxidation of the SH group. That is, it does not provide the SH group to move out of the protein and destroy the structure of the protein. So it maintains the structure of the protein, thereby allowing the enzyme activity to function normally. And it also helps in correcting the disulfide bond formation in the proteins with glutathione reductase. So in the presence of glutathione reductase enzyme, it helps in the correction of the disulfide bond that is formed between the SH and the protein in order to maintain the structure of the protein. In red blood cells, it maintains some membrane integrity and protects the hemoglobin from oxidation. So it forms a hydrogen peroxide, thereby allowing the membrane integrity. So the membrane does not rupture as such. Involved in amino acid transportation via gamma glutamyl Mester cycle. So it is involved in the amino acid transportation as well. It detoxifies the toxins and converts to mercapturic acids. So the toxins present inside the body is detoxified by the glutathione. It scavenges on the peroxides, which are the toxic forms of hydrogen and oxygen. So these peroxides are scavenged, that is removed, and a free radical is also released via glutathione peroxidase. It is an enzyme that is going to reduce the amount of toxic levels of peroxides by releasing the free radicals as well. Now moving to the second type of biologically important peptide is a thyrotropin releasing hormone. It is present in the thyroid, and the type of peptide here, it is tripeptide containing of 3 different amino acids. And where is it being released from the hypothalamus? This TRH is going to act on thyroid organ and release the thyrotropic hormones. It stimulates the anterior pituitary. So basically, hypothalamus is the master gland, and the pituitary is acting as its functional. So when hypothalamus releases the action that is performed by action that has to be performed by the pituitary. Now, pituitary acts as a stimulant and takes is acted by the stimulation of hypothalamus, and it releases the thyrotropic hormone. Now, oxytocin is another type of peptide where it is a nano peptide where there are 9 amino acids present in it. You can search for the 9 amino acids from NCBI website as well. You can find the structure of the peptide by going through the NCBI website, which is a biological database wherein you get to see the structure of the protein, the number of amino acids present in it, the genes that are coding the oxytocin as well. So where is this released? It is released in the uterus for the contraction when the when the pregnant woman is delivering a baby. So this oxytocin is from the source of posterior pituitary, and it stimulates the uterine contraction. Vasopressin, the ADH hormone, the adrenal, which is released by the adrenal glands. Here, the type is nonapeptide. Again, a 9 amino acids present in the peptide chain. The source is posterior pituitary, and the function of vasopressin is to promote the water reabsorption in the kidneys and increase the blood pressure. So here it is going to allow reabsorption of water in the kidney and maintain and sometimes increase the blood pressure as well. So vasopressin is said to be a flight hormone as well. Angiotensins. So these are of two types. One is angiotensin 1 and angiotensin 2. The angiotensin 1 has decapeptide, wherein 10 amino acids are present, and they are converted into angiotensin 2. So angiotensin 1 can be converted into angiotensin 2, which is an octapeptide containing 8 amino acids. This angiotensin 2 is more hypertensive, causes more hypertension. So the function of this angiotensin is to raise the blood pressure and stimulate the aldosterone release from the adrenal cortex. So angiotensin works on the blood pressure maintenance. So angiotensin 1 converts the angiotensin 2 into the functional form of peptide hormone. Now, methionine enkephalin is a type of pentapeptide with 5 amino acids present in them, and the location where it is released is brain, and the function of it is like opiate-like. It inhibits the pain sensation. It prevents the sensation of pain. Opiate is a type of a narcotic drug. Bradykinin or kelidin. Bradykinin is a nonapeptide, a 9 amino acid peptide, and kelidin is a decapeptide. So the function of this, they are potent vasodilators. They narrow the blood vessels, that is they constrict the blood vessels. They dilate the blood vessels, that is they widen the blood vessel. So the origin of this bradykinin and kelidin is plasma protein and activated by snake venom enzymes. So because of the potent vasodilation that happens because of the snake venom whenever released during the snake bite, the patient is going to have vasodilation. The blood is going to be flowing without any pressure, so it flows easily, and when that happens, there is chances of cardiac arrest and total collapse of the body organs and shutdown of the body. Peptide antibodies. Here the examples of peptide antibodies are gramicidin, vasopressin, thyrosidin, actinomycin. So these are some of the peptide antibiotics. So the function of all these is antimicrobial activity. So they are going to kill the type of microorganism they are going to act on. Gramicidin is going to act on the gram-positive bacteria. Vasopressin is going to act on the bacteria. Thyrosidin and actinomycin, similarly, they act as an antibiotic. Aspartame is a dipeptide which consists of aspartyl and phenylalanine are connected to each other by methyl ester. Now the property here, the aspartame is approximately 200 times sweeter than the actual sucrose, the natural sucrose or the natural sweetener that is obtained. So this can be used as a low calorie artificial soft sweetener in soft drinks. So you can see whenever you drink the Coca-Cola or the Pepsi, they are going to have a low calorie artificial sweetener which are going to cause increased sugar in the blood sugar levels, because these aspartame have approximately more than 200 times sweeter than the natural sugar that is obtained. The 10th property or the biological function is the gastrointestinal peptide hormone. Some of the gastrointestinal peptide hormones are gastrin and the secretin. These functions in regulating the gastrointestinal functions. The classification of protein is a topic under amino acid and protein chemistry. Here we are going to classify based upon the composition, function, shape, and with few of the examples. So proteins can be classified as based on functional classification into 9 types. So based on the function of the protein and the role in the body, we can classify them. So the structural protein provide the support and the strength. So few examples of structural protein are keratin, which is present in hair and nails, collagen, which is present in the bone. Enzymes are catalytic proteins. These speed up the biochemical reactions, such as some examples are hexokinase and pepsin. The transport proteins carry molecules within the body. Some of the examples that are involved in carrying molecules within the body are hemoglobin and serum albumin. So hemoglobin is involved in oxygen carrying. Hormonal proteins, they act as chemical messengers. Some examples are insulin, which are involved in regulating the blood glucose levels and maintain the blood sugar levels. The growth hormone, which is involved in the growth of the human body. Contractile protein helps in the movement. Some examples are actin and myosin, which are present in the muscle fibers. Storage protein, they store nutrients. Some examples are oval albumin, which is present in the egg, and glutathione, which is present in the plants. Genetic proteins associated with genetic material, nucleoproteins. Defense proteins protect the body, and few examples are immunoglobulins and snake venom proteins. Receptor proteins, they receive signals from the hormones or the viruses, and some examples are membrane hormone receptors. The classification based on chemical nature and shape is the second type of classification for the proteins. Here the proteins are divided into simple, conjugated, and derived proteins. Simple is made up of only amino acids. Under simple proteins, you can further divide into 2 types, which is globular proteins and fibrous proteins. So under globular proteins, these are spherical or oval shaped. They are soluble and digestible. Some of the examples of globular proteins are albumins, globulins, glutathione, prolamines, histones, globins, protamines, and lecithins. You can remember around 3 to 4 examples under globular proteins, because these are what are present on your body. So nothing is complicated unless you correlate it with your body and the surroundings that are present. Now albumins are soluble in water and dilute the salts. They can coagulate by heat. They can be coagulated by heating. Some examples of albumin are serum albumin and oval albumin. It's easy, you're just adding serum in front of the albumin. That is an example. Globulins, here solute in dilute and soluble in dilute salt solutions only. Some of the examples are serum globulins. Glutathione are soluble in dilute acids or alkalis. So you can remember, albumins are soluble in water, globulins are soluble in dilute salt solutions, and glutathione are soluble in dilute acids or alkalis. Prolamines are soluble in 70% alcohol. Gliadin, which is present in the wheat, is a type of globular protein. Histones are present in the chromosomes. They are strongly basic, soluble in water and dilute acid. Some examples of histones are thymus histones. The globins, Here they are non-basic, not precipitated by ammonia hydroxide. Protamines strongly basic, small and are soluble in ammonium hydroxide. Examples of protamines are sperm proteins. Lecithin, it is a carbohydrate binding and role in cell interactions. So examples are concanavalin A. Fibrous protein is a second type of simple protein. Here the fibrous protein, as the name says, it is long and fiber-like, insoluble and resistant to digestion. These are present in the plants mostly, and animals as collagen. And connective tissue lacks tryptophan, elastin found in elastic tissue, tendons and arteries, keratin found in hair, nail and horns, rich in cystin. Now, conjugated protein is a second type based upon the chemical nature and shape. The conjugated protein contains a non-protein prosthetic group. Here, the nucleoproteins, glycoproteins, mucoproteins, lipoproteins, phosphoproteins, chromoproteins and metalloproteins are present. Nucleic acid as prosthetic group, that is the group that is attached along with the regular protein chain. So for a protein chain, there is a conjugate or an additional group that is added. So these are called as conjugated proteins. In nucleoproteins, the prosthetic group is nucleic acids, glycoproteins carbohydrates, mucoproteins carbohydrates again, but the percentage is differing in glyco and mucoproteins. In lipoproteins, as the name says, lipid, and phosphoproteins with phosphoric acid, chromoproteins with colored prosthetic groups like in hemoglobin, wherein heme is the iron that is imparting the color. Metalloproteins are metal ions that are attached to the protein chain. Now, derived proteins is a product after breaking down or denaturating the protein. So some examples of derived proteins are primary derived and secondary derived. Primary derived is denatured or coagulated forms, whereas secondary derived is undergoing further hydrolysis of the products that are obtained from the primary derived ones. So under primary derived, the coagulated proteins are heated or chemically denatured. Some examples of coagulated proteins are the cooked egg albumin, and proteins is a early hydrolysis product that is insoluble. And some examples of proteins are fibrin from fibrinogen. Metaproteins are the third type of primary derived proteins, which is when further hydrolysis of protein takes place. The acid or alkali that is obtained on hydrolysis of a protein, that is called as metaproteins. Now, secondary derived is the further hydrolysis of the primary derived ones. So when the peptides, the proteins undergo continuous hydrolysis, you obtain proteoses, peptones, polypeptides and peptides. So these are the secondary derived proteins. Now, the nutritional classification of proteins based upon the essentiality of amino acid content for a human body, we can call it as complete proteins, partially incomplete proteins and incomplete proteins. So under complete proteins, it contains all the essential amino acids that is required in smaller amounts to the human body. Some examples are the egg albumin and milk casein. The partially incomplete proteins lack some essential amino acid, but they support moderate growth. So these are not essentially required, but they can provide certain amount of moderate growth into the body. Some examples that the protein that we consume are wheat and rice, which are consisting of low lysine and low threonine amino acids. Now, incomplete proteins are lacking one or more essential amino acids and they do not support the growth. So some examples are gelatin and zein. They have no tryptophan and lysine. Now, here is the summary of the classification of protein. This is a picture taken from the new Satya Narayan book on biochemistry. The proteins can be classified based upon the various methods, but here we have the uh, based upon the nature and chemical nature of the protein. Here we have simple, conjugated and derived. In terms of simple proteins, these are consisting of amino acid group only, and they do not have any additional group that is attached to the protein. The simple protein can be further divided into globular proteins and scleroproteins. So globular are albumins, globulins, glutalins, prolamines, histones, globins, protamines and lectins. Then scleroproteins are collagen, elastin and keratins. Conjugated proteins are the ones which have protein chain along with the additional group that is added to the proteins. Now nucleoproteins are nucleic acids. Glycoproteins have greater than 4% of carbohydrates. Mucoproteins have less than 4% of carbohydrates. Lipoproteins have lipids. Phosphoproteins have phosphoric acid. Chromoproteins have color imparting groups, and metalloproteins have metals. Now derived are the ones which undergo primary or secondary derivation from the protein. They can be either heated or hydrolyzed or coagulated. Now coagulated proteins are under primary, and proteins, metaproteins are the primary derived proteins. Secondary derived are proteoses, peptones, polypeptides and peptides. Now the function of the proteins is the final topic of today. So these are essential to understand why proteins are essential for a human body or any other organism that is present on the earth. The functions can be further classified as based upon the structure. Now the static. These proteins are required to maintain the strength and the framework in the body. Some examples are collagen and elastin, which are important to maintain the bone matrix, the vascular system and the connective tissue. Alpha keratin acts as a protein that gives epidermis, hairs and nails a structure and strength, and as well as framework. Now the dynamic is or the functional part of proteins. These are involved in varied cellular activities. So these proteins are going to act as enzymes, hormones, blood clotting factors, immunoglobulins or the antibodies, membrane receptors, storage proteins, genetic control, muscle contraction, respiration. So you can see the dynamic functional proteins are present in every nook and corner of your body that are maintaining the essential functioning of your body. So without these cellular activities, your body is never going to work normally, right? So thus protein is correctly called as building blocks of life. So these are going to be bodybuilders or the bodybuilder nutrients that are essential for your body, because they are providing all the cellular activities with the amino acids and function efficiently. Now these dynamic proteins are also called as the working horses of the cell, because they are doing their work 24/7, keeping the cell working as a efficient one. So here completes the protein as a whole topic. There are 3 different videos for protein and amino acid chemistry. All the 3 videos link shall be put in the description, and the notes of the amino acid and protein chemistry will be put into the link. If you did like the video, please subscribe to the channel and like it, and also share it among your friends and let the tribe grow. Thank you.